Chapter 26 deals with the nervous system infections. So once again, a brief little thing on anatomy. The meninges are the membranes that are surrounding the brain and the spinal cord. It's composed of 3 layers. You've got the dura mater, which is the outermost one, the arachnoid mater, which is the middle one, and pia mater is right up against either the spinal cord or the brain. CSF is cerebral spinal fluid that is flowing between the arachnoid and the pia mater. The central nervous system should be sterile, including that CSF. It helps with the blood-brain barrier. So sometimes if they do a spinal tap, what they are doing is removing the cerebral spinal fluid to test it to see if there is anything in it. But ideally, it should be sterile. Meningitis is inflammation of the meninges. Encephalitis is inflammation of the brain. So this is just a diagram showing, for review purposes, the layering of those meningeal layers. Neuropathies and numbness or tingling of the peripheral nerves. Peripheral nerves, remember, are any nerves that are outside of the spinal cord or the brain. Glial cells are supporting cells. Neurons are the ones that are capable of sending that electrical impulse. The myelin sheath is basically its insulation that's around the axon, the long extension from the neuron. The action potential or the electrical charge or electrical impulse is traveling down that axon. Synapses, that's the junction between neurons. It may be neuron to neuron, it could be neuron to muscle, neuron to gland. Neurotransmitters are the chemicals that are being released from one neuron, and it goes through the synaptic cleft, which is the space between the neuron and what it's connecting to, which could be another neuron or a muscle. The neurotransmitter is going to activate some type of a response. And so here you see your classic neuron. It's got the large cell body here. These little colored structure, that's the insulation. Those are specialized. In this case, it's in the central nervous system, showing the oligodendrocytes, which are specialized cells that are forming this insulated sheath around the axon. So the electrical charge would be traveling in this direction. Right here is your synaptic cleft, that space between your presynaptic neuron. So the impulse is coming in this direction. It's going to trigger the release of the neurotransmitters, those chemicals, which flow across the space here and will bind to receptors on the postsynaptic neuron. And then that triggers the response. So if we start off once again looking at bacterial infections, Neisseria meningitidis is a gram-negative. It is a causative agent of meningitis. Very serious disease. You sometimes will see death within hours of the onset of symptoms. So if you suspect this, you need to start treatment immediately. Highest threat is among infants, adolescents, and young adults. It can be transmitted by respiratory droplets. It can be transmitted by direct contact. This is one of the concerns that we have on college campuses. You have a lot of young people. You have them often, we pack you guys in classes or in dorms, or you're in confined spaces. And so you're increasing the chances of this contact in order for the transmission to occur. To prevent it, there is a vaccine. You can treat with antibiotics, but there's obviously a time frame. You need to treat immediately. This is showing over here on the far left. This is a normal brain during autopsy, and over here on the right is the brain of a patient who did die of bacterial meningitis. So this membrane right here they're pulling away is a dura mater, and there is some pus that is underneath there. So you can definitely see the difference between the two brains and visually what they look like. Neisseria meningitidis. Neisseria, oftentimes, it is a coccus. It often is in pairs. Sometimes they refer to them, we mentioned this previously with the Neisseria gonorrhea, that they kind of look like kidney beans. So they're in pairs like that. So like I say, a lot of colleges and universities now do require that incoming students be vaccinated against meningitis. It's safety concerns, obviously. Protection for you. Streptococcus pneumoniae is gram-positive. It can also cause meningitis. It also can be transmitted by direct contact as well as by aerosols. A vaccine is also available for it. If someone does contract it, you can treat it with antibiotics. This over here on the left is fluorescent antibody, which is why it's glowing green. That is Streptococcus pneumoniae that was extracted from the cerebral spinal fluid. And over here on the right, it is growing on a blood agar plate. Haemophilus influenza type B, we often abbreviate that as HIB. This is gram-negative. It is also another causative agent of meningitis. It can be transmitted by inhalation of aerosols or by direct contact. There is also a vaccine against, it's the HIB vaccine. You can also treat with antibiotics. Streptococcus alcalacea is gram-positive. This can cause neonatal meningitis. This is if the mother is infected, the newborn can be infected during a vaginal birth. So to try to prevent this, you can either do C-section, give the mother antibiotics via IV during labor, and then you can treat the newborn with antibiotics as well. Clostridium tetani. Clostridium is gram-positive. It is anaerobic, and it is an endospore forming. It's commonly found in the soil. It causes tetanus. A common term for tetanus is lockjaw. Clostridium tetani produces a neurotoxin. It can be transmitted a couple different ways, but if you have a wound, and then it can penetrate into that wound. So you might be thinking, well, if you have a wound and it's transmitted that way, why is it not considered with the skin? That's the port of entry, but the effect is on the nervous system. There is a vaccine, the tetanus vaccine, that is available, and if somebody contracts it, you can give them an antitoxin, because the effects are due to a toxin that is released by Clostridium. Clostridium tetani as well as Clostridium botulinum, which we're going to talk about next. Being anaerobes, I don't know if you guys were ever raised being told, you know, if you're shopping and you see a canned food item and it's bulging, don't buy it. There's truth to that. In the canning process, sometimes spores may get in there. The concern with Clostridium is that because they do produce endospores, they can survive. Is that inside the can, as it becomes anaerobic, that favors now the environment for Clostridium, which is an anaerobic organism, to grow. And then as it's growing, it can be releasing the toxins. And then you buy the food item, and did you heat it up hot enough to destroy that toxin? Well, I don't know what your cooking methods are. You might not, and then you can get sick from eating it. So if the can is bulging, just get another one, okay? This shows someone that is suffering from systemic tetanus. What happens is you have sustained muscle contraction. The toxin affects at the neuromuscular junction where the nerve and the muscles connect, and it triggers the muscles to contract, and they never relax, and so all these muscles are contracting. Clostridium botulinum, same genus, gram-positive, endospore, anaerobic, also commonly found in the soil. It is a causative agent of botulism. There are different forms of botulism. There's wound, infant, and adult intestinal. Wound, where it enters once again through a wound. Infant, one of the problems here is sometimes infants get it. Honey is a source, unfortunately, at times for it. Adult intestinal, usually you have ingested it, and that's how it's transmitted. It also produces a neurotoxin. The neurotoxin that is produced by Clostridium botulinum is the most toxic natural compound known to man. Just think about that for a sec. It's transmitted by ingestion of either the toxin or the bacteria in contaminated food, or as I said, the bacteria enters the wound. In terms of prevention of this, do not give honey to infants under 1 year of age. That will prevent the infant botulism. Heat your food up. Once again, don't buy a bulging can. If someone has botulism, you can treat it with antitoxin. There are some antibiotics you can also use to treat as well. Both the botulism and the tetanus, these toxins, they both work at the neuromuscular junction. Before we continue on, let me just say that the tetanus, the neurotransmitter that is released and binds to the muscle that starts the muscle contraction, it is never broken down, which is why you have sustained muscle contraction. For botulism, the toxin works almost in the reverse way. You never release the neurotransmitter in the first place, so you never ever get the release of the neurotransmitter, so you never get a muscle contraction. That's one of the reasons why they use it for cosmetic reasons sometimes. Prevents wrinkles because you never have the muscle contractions. Just keep in mind that for tetanus there is a vaccine, and it's important to get the boosters. They keep changing over the years how often you need the boosters. It was 10 years, then they went to 5, then they went back to 10, then they went to 7. So just check on it. They keep changing it so much it's hard to keep up. And by the time you guys are out working in the field, they probably will have changed it again. For botulism, like I said, they can administer antibiotics and antitoxin for it. Just be careful if you plan on getting Botox treatments. It's a diluted form of the toxin. Just make sure the person has a steady hand, they know what they're doing, and think about what they're doing beforehand. Listeria monocytogenes gram-positive causes listeriosis. This is transmitted by ingesting contaminated food. Processed meats and cheeses, fresh fruits and fresh vegetables are notorious for having it. Most people may just have a mild case if they're symptomatic at all. One of the big problems is for pregnant women, because it can cross the placental barrier and cause adverse effects on the developing fetus. Newborns, elderly, and immunocompromised are also going to be at higher risk. It seems lately we have had some, across the U.S., recalls of different food items like processed cheeses, processed meats that have been contaminated with listeria. So this is a concern. It is not something that happened in the past. It is currently a concern. Just be careful with it. Treat with antibiotics. So this is just showing the listeria organism as it's starting to infect a host over here. Just be careful. They will sometimes recommend for a pregnant woman not to consume processed meat or processed cheeses. Mycobacterium leprae. This is an acid fast. In the Mycobacteria genera, it does not take the gram stain very well because of the high lipid content with the cell wall, so it's an acid fast one. This is the causative agent of Hansen's disease, which was formerly known as leprosy. Several years ago, they decided to change the name of leprosy because there's a negative social connotation with leprosy historically. To me, the problem has always been they switched it to Hansen's disease. But whenever you hear someone or read someone about somebody calling it Hansen's disease, it's immediately followed by formerly known as leprosy, because nobody knows what Hansen's disease is. So I'm not real sure how effective changing the name has been, because now everybody lists both names. This seems to be transmitted by inhalation, but it has to be very long-term exposure. Most people have natural immunity against it. There's only like 5% of the population globally is even susceptible to it. There have been many recorded instances of where individuals who have had long-term exposure with someone who has been diagnosed with leprosy and they never get it. It prefers cooler temperatures, so that's why it usually affects the extremities, the fingers, and the hands. To this day, we cannot grow this bacteria in a pure culture in lab. We can grow it in armadillos and we can grow it in the foot pads of mice, but we cannot grow it in pure culture. There are drugs that we can use to treat the disease. In terms of what it does is it will arrest the disease. That means it will stop it at that point and it will not progress any further, but it is not a cure. So ideally, if you can diagnose someone with leprosy as soon as possible, start the treatment, and you will not have any additional damage occurring. There's misconceptions where some people say, oh, well, the fingers or the toes fall off. No, they do not. They get reabsorbed. And so the fingers may end up looking like stubs. Now that we have these drugs that we can start treatment as soon as possible, hopefully we won't be seeing that as much anymore. There's often nerve damage. I had the opportunity to tour a facility in Louisiana. It was in Carville. It was run by the Department of Health at the time. That was the World Center for Research for Leprosy. And there were people who were living there. At that time, they had the option, if they were diagnosed, of living there. If they were single, they could live in these little like dorm apartments. There was quite a bit of testing and education that went along with it. They had occupational therapists to help teach them, because there was often nerve damage in the hands. If you're cooking, then you always put on an oven mitt, always. Whether you think something's hot or not, you grab it with the oven mitt on from both sides. The week prior to us going for our tour, they said there was one of the residents who ended up burning his hand because he went to go pick up a teapot and didn't realize it was hot. He had no feeling in his hands, and the only way he knew it was burning his hand was when he could smell the skin burning. Sometimes there's problems walking. They had a shoemaker there who all of the shoes that he made, and this was in 1983, so before Velcro was popular on shoes. It didn't even exist at that point. And he was already using Velcro because some of the residents had problems with the dexterity, with the movement of their fingers. They couldn't tie shoelace. And so he developed using the Velcro. The shoes had a little bit of, they didn't have a flat sole. It was made so that they would rock forward to naturally take the next step, because some of them actually had problems if you can't feel your feet. And that's going to influence the way you walk. So it's very interesting to see how they just adapt it. Depending on the individual's condition, they adapted so they could still do everything. I remember our tour guide was this pleasant woman who said, I can do anything you can do. I just might do it a little bit differently. But it's not going to stop me. So Mycobacterium leprae, like I said, it's one of the few diseases that the government, they're used to monitor everyone in the world, because like I said, it was the World Center. So they kept track on everyone in the world who had ever been diagnosed. And at that point, it was the only disease that the government would completely pay all of your treatment costs for. So this is showing on the left here, this is the nose of a patient. You're starting to see reabsorption occurring here. And over here on the right is right in here. When you do an acid-fast stain, the acid-fast bacteria will look red and all the other cells will look blue. Moving on to viral infections, encephalitis. There are several arboviruses. They're all single-stranded RNA that can cause this. Mosquitoes are going to be the vector. So transmission is from, there's another animal host, the mosquito bites it, then it comes and it bites the humans. There's 4 main types. They have different hosts. They tend to be species specific for what type of mosquito, what species of mosquito is the vector. There is a vaccine that is available, but for only one type. You treat with supportive care. And this is just showing some pictures associated with examining tissue samples, looking for the infection, and then over here on the right is looking at CT scans on the left over here, and then on the right here is MRI scans of the brains of individuals with encephalitis. The Zika virus. This is a single-stranded RNA virus. The mosquito is a vector. It's transmitted between humans. So one human's infected, mosquito bites you, then it goes and it bites somebody else, and that's the way it's transmitted. One of the concerns is certainly with pregnant women. There have been found to be abnormalities in the developing fetus, so that's why pregnant women are very high risk. You can treat with supportive care. Women who are pregnant, when they become infected with Zika virus, the Zika virus can pass that placental barrier, which is why the fetus is at risk. Not always, but there does increase the chance of the woman having an infant, a newborn, that has microencephaly. This deformity basically is smaller head and smaller brain. Rabies. Rabies is another single-stranded RNA virus. It's transmitted by a bite. So you have an infected mammal. It's with mammals or the other host, and then that infected animal bites a human. If it is not treated and progresses, it causes encephalitis. One thing about rabies, once symptoms appear, it's too late. Over all of recorded history, we only have 5 reports of someone who, once they showed symptoms, that they survived. Those odds are not in your favor at all. Prevention. One thing that has been huge is to vaccinate domesticated animals. Vaccinate your cats and your dogs. The other big thing is don't handle wild animals. Wild animals are wild animals. Leave them alone. Several years ago, I believe it was up by Tyler, where there's some students that were on campus, and they picked up, one of them saw, oh, isn't this cute? Look at this cute little thing, and picked it up. It was a bat. Okay, number one, a bat should not be out in the middle of the day. That's abnormal behavior. When you see abnormal behavior in animals, it usually means they're sick. Leave them alone. Definitely. Okay, don't touch any wild animal, but especially if it has abnormal behavior. They picked up the bat. There were 4 of them, and they were passing them around. And then a 5th person came up, and they're like, oh, look what we saw. Isn't this so cute? And when that individual saw it, they're like, no, that's a bat. You shouldn't be touching it. They're a mammal that can carry rabies. Suggested that the ladies turn the animal in, which they did. It was tested, and it came back positive for rabies. Because when they were handling it, they got some small bites, scratches, etc. It turns out all 4 of the ones that were handling it had to be tested and treated for rabies. So you can receive shots if you have been bitten by a suspected rabid animal. You need to start the shots right away. Don't wait to see whether the animal shows the signs of rabies or not. Because if you wait too long and then there's a delay before you start showing symptoms, it might be too late for you. So you have to start the series of shots. But like I say, the easiest thing is vaccinate domestic animals. Don't handle wild animals. The vaccine that's given is a weekend vaccine. Now, there's not a vaccine for the general public. There is a vaccine that is available for rabies. It's for select groups. Years ago, I can remember touring a state scientific lab for the state of New Mexico. And they do have someone, part of the position I was looking at would have been, they have one person that's designated that does all the rabies testing. So when there is a suspected case of rabies, the animal, which hopefully the person has been able to identify, to positively identify it, the animal needs to be sacrificed, because the only way you can confirm rabies is to test the brain tissue. So you're going to have to sacrifice the animal. At least in the state of New Mexico, the individual who was responsible for doing that autopsy and testing for rabies on the animal, they did receive a rabies vaccine for protection. This is showing here, you can see the rabies virus here. They look like little bullets. Here. Poliovirus, single-stranded RNA. It is the causative agent of polio. The full name is poliomyelitis. That is transmitted by the fecal-oral route. You can have contact with droplets or aerosol. Prevention. Polio used to be a huge concern. Since vaccines became available, the number of cases has dropped dramatically. It has been such a good success rate when talking about vaccines. There were two vaccines that were developed pretty much at the same time. The Salk vaccine, which is an inactivated virus, and then the Sabin vaccine, which is an attenuated virus. That means it's a weakened virus. Both were equally successful. The attenuated virus, just because it's a weakened virus, there's a very minor, small chance of contracting a mild form of the disease. It is no longer used in the U.S. You treat with supportive care. Thank goodness, as I said, the number of cases have been drastically reduced. The hope has always been that polio would be another one of the diseases that would be eradicated globally. Unfortunately, it is not. It is still in areas where there's military unrest, political unrest, and so it's not safe to get healthcare individuals into those areas to vaccinate everyone. There's also starting to be a concern amongst individuals who are against vaccinations, where they are not vaccinating themselves or their children. They are putting them at risk now. They're starting to see a little bit of an increase in polio in some of those individuals. So this is shown, this was discussed in a previous chapter. This is an iron lung treating someone that had polio where they are not able to breathe unassisted. And then over here you're showing someone that has some leg braces on to help them walk. There's a wide range of symptoms of polio. Some can be very minor, some very severe, as you can see with the iron lung that was needed. There's no way of knowing ahead of time which way an individual is going to react. So this in the far left is showing the polio virus, what it looks like. And then over here we have the two individuals that developed the vaccines. We have Dr. Albert Sabin here and Dr. Jonas Salk here. Like I said, they both developed the vaccines right about the same time. They've both been the vaccines have been very, very effective, very successful. Just kind of a side FYI, I was listening to a podcast fairly recently, and it was a virologist who was talking, who had met both of these individuals. She knew both of them personally. And something that I had never heard, which was kind of in some ways sad here, both of these men, like I said, very successful. Both of them have always been recognized for their contributions for developing. This was huge to get the development of this vaccine to prevent and decrease the number of cases of polio. Unfortunately, I don't know if it's egos, pride, whatever, but according to this woman who knows both, has worked with some of them, said, unfortunately, these two gentlemen absolutely hate each other, which is really sad to see, to the point where she said if one of them is in the room, the other one will leave. And if he can't leave the room, he'll go to the furthest corner away. And it's like, that's really sad. We're just trying to help people. So it's sad that the egos get in the way of things like that. Sometimes adults still act like kindergartners. Prion infections, transmissible spongiform encephalopathies. Prion is an infectious, abnormal protein. The folding of it is abnormal, and that causes it to become infectious. It can infect the brain, causing degenerative, fatal neurological diseases. When you look at the brain tissue, it becomes much more, there's a lot of open spaces in it, it's breaking down. It looks like a sponge, which is why we call it spongiform. When it's in sheep, it's scrapie. When it's in deer and elk, we call it chronic wasting disease. In cattle, it's referred to as mad cow disease. In human, it's Creutzfeldt-Jakob disease or Kuru. So how is it transmitted? You're either exposed to an infected animal, or usually what happens is you're exposed and ingest contaminated meat. So you ate meat from a cow that was infected, or you ate venison or elk from an animal that was infected. You can give supportive care. Unfortunately, as I said, it's degenerative. It's just going to continue and progress and get worse. And unfortunately, at this point in time, there is no cure. There's a lot of research right now trying to figure out. We finally identified what it is, what's causing, like mad cow disease. It was happening. What causes it? Well, that's when we discovered prions. Okay, fine. Now we know what the causative agent is. That gives us help in trying to move forward. But where the research is now is trying to figure out, can we get a cure? This is just showing how you have conversion or changing of the proteins. For fungal infections, Cryptococcus neoformans. This can cause meningitis. It's commonly found in the soil and in bird droppings. It's transmitted by inhaling those fungal spores. Individuals that are at high risk are going to be your immunocompromised. So you treat with antifungal drugs, and you're going to have to treat long term. Usually fungal infections, the treatment certainly is longer than for, say, a bacterial infection. And so this is showing the spores, the yeast cells here. They do have this large capsule around them that helps to protect them. Other fungal infections that cause meningitis. Usually this is seen only in immunocompromised individuals, but there are several different types. These include Aspergillus, Fumigatus, Candida albicans, Coccidioides immitis, Cryptococcus neoformans, Histoplasma capsulanum, and Rhizopus aureus. All of these you can treat with antifungal drugs, and once again, it's going to be a long-term treatment, or certainly longer than what you would see with bacterial infections. Moving on to protozoan infections, Naegleria fowleri. This is a type of amoeba flagellate. It's in soil and water. It causes a type of meningoencephalitis. It's transmitted usually because somebody is swimming in contaminated fresh water, and the protozoan is in that fresh water, and you swallow or inhale some of the water. This, unfortunately, the disease progresses very fast. Now, there is some experimental drugs they're trying to use to help treat it. This is the organism. I don't know how many of you have heard of it before, but sometimes, usually in the summer, because it's when people are swimming in and lakes, where you hear of an individual where they went swimming and 24 hours later they die, and they're like, well, what happened here? It's oftentimes this protozoan. It seems that we've had more cases of it in the past few years. And that always begs the question, are the number of cases increasing actually, or is it because our diagnosis is better? Either way, hopefully this new experimental drug is going to work. And this is what it looks like here. You took a sample from a tissue sample from a patient. Amoebic encephalitis, amoeba in fresh water, just a couple different species. As you can see, that are causative agents here. Once again, it's transmitted by exposure to the contaminated water. If you have a break in the skin, sinuses, that's how it's able to get in. You can treat it with antimicrobial drugs. And this is, as you can see, samples of the necrosis, the dying tissue that has occurred right here, and then you've got the close-up right here that's occurring in the brain. For some of these with the water, if nothing else, it may trigger you to keep your mouth shut when you're swimming. Trypanosoma brucei. This is often referred to as African sleeping sickness. This is also protozoan. It's a hemoflagellate. The vector is a tsetse fly. Prevention, control the vector. Use netting around, the bed when you're sleeping at night, etc. If you do contract it, you can treat it with antimicrobial drugs. It's most effective in the very early stages. So once again, early diagnosis is always best. And this is what it looks like in a blood sample. Toxoplasma gondii, cats are the definitive host. That's where they prefer to live. And this is, we've talked about this before, cats are going to become infected with the protozoan. Protozoans, once again, they go through their life cycle. They have different stages. And so in the cat feces, they shed the cyst form of it. And then oftentimes, say if you're cleaning the litter box or something and dust is raised up, you can inhale those cysts. You can also have it transmitted by ingestion of infected meat. Pregnant women, immunocompromised individuals are at the highest risk. So don't handle, if you're in the high risk group, don't handle the cat litter. You can treat with drug therapy, the use of several different drugs at once on it. This is what the Toxoplasma gondii cyst looks like. Moving on to helminthic infections, Taenia solium. This is a pork tapeworm, causes neurocysticercosis. It is transmitted by ingestion of contaminated food or contact with contaminated surfaces that you ingest the cyst form and then proceeds with the whole tapeworm. Treat with antimicrobial drugs. In some cases, you may have to do surgery to remove the cyst. And this is a brain scan showing several of these cysts that are present here from the infection. So this is the final chapter for this course. These last several ones talking about diseases. If nothing else, hopefully you remember clean water, clean food, cook your food. That is going to do so much to prevent the transmission of diseases. Vector control, control the mosquitoes, control the ticks, control the fleas. If you have to put netting up around bedding, depending on your location and the situation, do what you can to prevent that transmission. If you can break that transmission between another host or the vector and you, then you're going to prevent transmission of the disease. Number of cases go down. As I said earlier once, yes, and here we're looking at this organism causes this disease. In other classes like pathophysiology is where you look at more detail. What are all the symptoms of the disease? How does it progress? And specifically, at that point, you can look at what specific drugs you use for treatment. This is an intro class. We focus on medical microbiology, but we've covered a lot of different things, including immunology and just some of the basic from genetics. I don't know if this course ended up being what you thought it was going to be. Hopefully you've learned a lot from it. Hopefully you've enjoyed it along the way. One of my concerns is always students usually start microbiology with the fear that, ooh, all of these germs are out here to kill me. Hopefully by now, you know, at least in front of me, don't call them germs. And hopefully you remember that of all the microorganisms that are out there, that only 5% of them are human pathogens. And that means the other 95%, they're beneficial to us or they're just there. But they're not all out there to kill you. And very early on, the first day I had mentioned in chapter 1, and I'm going back to that now just to say, you know, if you stop and think about it, and unfortunately, when we're focusing on people going into the medical career, we focus on the medical bacteriology, or especially bacteriology, but certainly medical microbiology aspects. But remember, you cannot go through a single day without the beneficial aspects of microorganisms, be it that they're breaking down our waste products. On a more enjoyable thought, we wouldn't have bread without microorganisms. We wouldn't have cheese without microorganisms. We wouldn't have a lot of our food products without microorganisms. And so they're not all bad. So just kind of think about that. I hope you have learned something from this. I hope you've been able to take away some positive things from this. And I hope it wasn't quite as scary as what you maybe were afraid it was going to be. And recognize that, yes, there is this whole world out there that we can't see, but it doesn't have to be scary. I had someone, actually it was a relative, ask me as the COVID pandemic was just starting, and she obviously knows what I do, and she asked me early on, she goes, Paula, are you scared of COVID? And my response to her was this: I'm not scared of it, but I have a respect for it. And to me, that's a healthy way of looking. Should you be scared of all of these microorganisms? No. The majority of them, frankly, really don't care about us. It's such a small group that are pathogens. Are some of those pathogens deadly? Yes, they are. But if we just have a healthy respect for it, you don't have to have a fear of it, but you respect it. So good luck as you continue on after this course, and whether you're using microbiology a lot in your future career or maybe just a little, I hope you, like I said, had a little bit of fun and learned a bit. So good luck to all of you.